Hello, my name is Draghici, and I'm doing my presentation on human cloning. Envision someone in your life suffering from heart disease or liver failure, and the only way to help them out is to have an exact replica of the organ. Today, this is a major issue we face because the only way to accomplish this is through a process called cloning. This process, along with stem cell research, are exciting in the fact that our advancement in technology and science are at an all-time high, but happens to be a very controversial issue worldwide. Today, I will speak with you about the history of cloning, the process and types of cloning, and the pros and cons to cloning. The history of cloning dates back to the year 1901 when a scientist by the name of Hans Spemann split a two-celled newt embryo into two parts, successfully creating two larvae. Nearly 40 years later, Hans concluded that cloning was possible, and he theorized that animals could be cloned by fusing one embryo and one egg cell. Francis Crick and James Watson discovered the double helix structure, which is very important to cloning because they realized that you can separate DNA into two separate eggs. In the year 1996, Dolly the sheep was born. Dolly was the world's first mammal to be cloned from an adult cell. The birth and survival of Dolly was not only one of the most significant scientific breakthroughs ever, but also proved that cells previously found in one adult can be transferred from one being to another. All was well with Dolly until 6 years later when she had premature arthritis and progressive lung disease. Many believed this was due to having already aged cells. Over Dolly's life, she had 4 children that she was able to prove that cloned humans or animals are able to breed. The fact that Dolly ever existed is a life-changing event. Now, after learning about the background, we will talk about the different types of cloning and how these processes work. The first type is called gene cloning. This process takes DNA from a single organism with a gene of interest and cuts the gene into pieces with restriction enzymes. These enzymes read the nucleotide sequence of the DNA and recognize specific sequences. These enzymes cut the DNA by breaking the bonds in the DNA strands as well as bacterial plasmids. These are then placed into a test tube, and the enzymes pieces will mold with the plasmids to create a new combination of DNA. The new DNA is then transferred into bacteria by heat shock. This is the most popular type of cloning at the National Human Genome Research Institute. The next type of cloning is reproductive cloning. The first thing scientists do is remove a somatic cell from the certain organism they desire to make a copy of. Then, the DNA is transferred into an egg that also had its nucleus removed. The first way is to remove the nucleus of a somatic cell with a needle and inject it into the empty egg. From there, it is implemented into the new organism. This process was used in creating Dolly the sheep. The last type of cloning is therapeutic cloning. In this process, there is no sperm fertilization involved in the process, as well as no implantation from an egg. The scientist then removes the nucleus of a somatic cell using any other body cell other than an egg or sperm cell. The nucleus that was removed from the somatic is placed into the egg. The egg, containing all genetic material and information needed, is then stimulated and creates a blastocyst, or a cluster of cells. Now I will talk about the pros and cons of cloning. There are a lot of pros or good things that come along with cloning. The first one being is that as humans age and reproduce, the DNA lines from the parent to the offspring become damaged, and they get more mutations and defects that are decreased when you use a clone, because they're not naturally reproducing. Another benefit of cloning is the traumatic injury time. If someone has a organ or something that they can transfer directly into them that matches what they previously had, it's easier for the body to accept it and for them to recover faster. Cloning can be used to replace failing organs and help decrease the number of people needing transplants, which is a massive issue today because a lot of people die or have to wait a long time to get transplants. Cloning of world-changing human beings would be immenseful because there would be a lot more people that are helpful in the world than that do harm. Some parents are unable to have children, so this is a great way for those parents to somehow make an emotional connection with someone that could still be a part of them. And this is the same case for the same-sex marriage. Now, onto the cons. There are a lot of disadvantages to cloning, some of which take away from the uniqueness of the individuality of the person. Even though a clone is creating a brand new life, there is a loss of individuality because it is simply creating a twin of another person. Another con is that these cells can lead to premature death. Some parents dislike the child they receive, so this way they are able to construct their own child in a sense, and that is not right because it's not natural, and people should not be able to, in a sense, create a robot or a perfect child. So the three main topics we covered today were the history of cloning, the process of cloning, and how the process occurs, and the pros and cons of cloning. In a world with many clones, life as we know it would be completely different. The clones would be treated as more parts than as an actual human being. Anticipate someone needing a transplant to save their life. The clone would be used to give that person the transplant because they're a clone. They are not naturally born. It is almost like when things go wrong with machinery, they use other parts to fix more important parts. In this case, that would be natural human beings and the people that have a lot of money. Cloning represents a very powerful and immediate example in which we are in danger of turning procreation into manufacture. Thank you. That was so bad.
Yeah, you were reading off the paper the whole time.
I
wasn't
don't-
even paying attention.
I don't know it.